Assalamualaikum. We will continue part 4 of the chapter 10 lecture, Development of Female Gametophytes. So this figure illustrates the development of a female gametophyte or the embryo sacs. The embryo sacs develop from the megaspores within the ovule. So within the ovules, megasporangium, there is a large diploid cell called the megasporocyte. This is the megaspore of the mother cells. And then the megasporocyte divides by meiosis. Meiosis process that will give rise to 4 haploid cells. But in most species, only one of these 4 cells survive as the megaspore. And then it will undergo mitosis 3 times. The megaspore will undergo mitosis 3 times to form the embryo sac, a multicellular female gametophyte. And then the ovule now consists of the embryo sac along with the surrounding integuments, which is the protective tissue. So this figure further illustrate the development of female gametophyte from the embryo sac mother cell, which is diploid, and then undergo meiosis to form 4 haploid cells, and one of it will become megaspores. And it will undergo mitosis 3 times, first mitosis, second mitosis, and the third mitosis, and then this will form 3 antipodal cells, 2 polar nuclei, 1 ovum, and 2 synergids. So this is the becoming, after nuclear fusion, this will become mature embryo sac, which is the female gametophyte. Just before fertilization, 7 nuclei present, 6 are haploid, and 1 is diploid. So within the ovule's megasporangium, there is a large diploid 2N cell called megasporocyte, megaspore of the mother cell. And then sporangium is an enclosure in which spores are formed. Megasporangium is a sporangium that produces megaspore that give rise to female gametophyte or the embryo sac. And then this megasporocyte divide by meiosis, which will then form 4 haploid megaspores, in which only one of the megaspores survives, the other 3 will degenerate. And then the single surviving megaspores in the ovule develop into an embryo sac, which is the female gametophyte. The megaspores undergoes 3 mitotic division, so 3 times mitosis to produce 8 haploid nuclei, which are enclosed within 7-celled embryo sac. The 8 nuclei arranged in a precise position. One of the nucleus is located near the opening of the embryo sac in the egg cell. 2 located in a single cell in the middle of the embryo sac, which are called polar nuclei. Another 2 nuclei contained in the cell called synergid. So these 2 nuclei flank the egg cell. The other 3 nuclei reside in the cell called antipodal, located at the end of the sac, opposite of the egg cell. Following pollination, a process called double fertilization, in which one sperm cell nucleus fuses with the egg cell to form diploid zygote, and the other sperm cell nucleus fuses with 2 polar nuclei. This will form triploid endosperm nucleus, and then the seed development will begin. So this figure shows the formation of pollen grain, which is the male gametophyte, and the formation of embryo sac, which is the female gametophyte. For the pollen grain, in the anther, the microsporocyte, the diploid microsporocyte undergo meiosis to form microspores and then undergo mitosis to form pollen grains, which include generative cell and also tube cell. And then when it arrives the stigma of another flower, it will turn and form pollen tube to deliver the sperm to the egg. And then for female gametophyte development, the megasporocyte will undergo meiosis to form 4 megaspores, in which only one of it becoming the surviving megaspores and undergo 3 times mitotic division to form antipodal cells, polar nuclei, egg cell, and synergy. So this is another figure illustrating the same information, which is the development of male gametophyte and female gametophyte. So for comparison of asexual and sexual reproduction, so in terms of number of organisms involved, in asexual reproduction, only single parent is involved. And in sexual reproduction, it requires 2 unisexual parents, which is 1 male and 1 female, or hermaphrodites. Hermaphrodite is a combination of both male and female. And then involvement of sex cells. So in asexual reproduction, there is no gametes produced. In sexual reproduction, 2 types of gametes, sperm for male and ovum or egg for female, produced by the parents. In terms of cell division, in asexual reproduction only involve mitosis, while in sexual reproduction, it involves meiosis to produce gametes and mitosis in the development of zygote. So in terms of genetic variation of the offspring, in asexual reproduction, the offspring is genetically identical to the parent, and in sexual reproduction, the offspring shows genetic variation. So the genetic construct of the offspring is unidentical to the parents. The number of offspring in asexual reproduction usually large and produced in a very short time. And in sexual reproduction, is usually comparatively less number of offspring produced. So in this figure, it shows mode of asexual reproduction, which is the vegetative parts, the stolon or runner. And this figure shows sexual reproduction, which involves sporophyte phase and gametophyte phase throughout the lifetime. For pollination, pollination can be divided into 2, self-pollination and cross-pollination. Self-pollination occurs within the same flower between different flowers on the same plant. So it involves only single plant. It can be within the single flower or between 2 flowers, or more than 1 flowers within the same individual, which is the plant. And then cross-pollination occurs between the different individual plant of the same species. So the flowers is between the 2 individuals of same species of plant. Pollinators are organisms that carry pollen grains from the stamen to stigma. So the pollinators can be insects such as bees or other animals such as birds or bats. The majority of pollinators are biotic agents. Biotic means living things, living agents. Bees are the most frequent and characteristic pollinators of flowers. An insect usually attracted by odors of the flowers, while bird-pollinated flowers are characteristically odorless and red, with the nectar not readily accessed by insects. So, bird can access the nectar of the flower. For example, hummingbird. And other plant species can be pollinated by abiotic agent or agent that is not living things. For example, wind and water. A characteristic of insect pollinated flowers. So the flowers are usually large, brightly colored and scented. This is to attract the insects for pollination. If the flowers are small, they may group together to form conspicuous inflorescence. This is a type of flower distribution. And then nectar is often present to attract the insect, and pollen is fairly abundant. So the pollen grains are light, sticky and heavy, usually with rough surfaces. So the pollen can easily cling to the body of the insects. And then stamen may not be pendulous, and stigmas are usually not feathery and do not protrude. And the stigmas are sticky, so that pollen grain can be settled on the stigma and not easily displaced or removed. The nectar guides may be seen on the petals. For the characteristic of wind-pollinated flowers, so these flowers are pollinated by wind. So the flowers are usually small, dull-colored and scentless. So this is because it does not need to attract insects. The nectar usually absent also because it does not involve biotic agent. Pollen is abundant because wastage is higher. So in the process of wind pollination, more pollen is wasted. So pollen grains are small, dry, smooth and light so that they are buoyant and easily blown by air currents. The stamens usually have long, slender filaments that sway in the slightest wind, and the pollen grains are hence shaken out from the dianthus. The stigmas protrude and are large and feathery so that they provide a large surface area to catch the pollen floating in the air. And nectar guides are absent in the flowers. So see how this is dandelions. It is very feathery, easier for wind pollination. We will discuss on a mechanism that prevents self-fertilization in part 5 of the lecture. Thank you.